My name is Tanir Barak. I'm a medical doctor and scientist, and I'm working with Professor Murat Günal in Department of Neurosurgery, Yale School of Medicine, and I will be talking about the genetics of intracranial aneurysms in this presentation. Stroke is, following the ischemic heart disease, is one of the leading causes of mortality and disability worldwide. And according to 2019 American Heart Association Heart Disease and Stroke Statistics, every 42nd someone in the United States has stroke. While nearly 87% of the stroke cases are ischemic, approximately 13% are hemorrhagic, including the subarachnoid hemorrhage cases. And among the different types, subarachnoid hemorrhage due to a ruptured intracranial aneurysm carries the highest mortality, with nearly 1 in every 4 subarachnoid hemorrhage patients die suddenly before being admitted to hospital. And although IA rupture is rare, it is responsible of 500,000 hemorrhagic strokes worldwide every year. IA prevalence increases with age and affecting approximately 3 to 5% of world population. And given the very poor prognosis of the aneurysmal subarachnoid hemorrhage, diagnosis of aneurysms prior to rupture is very important, since surgical endovascular repair can prevent morbidity and mortality. While environmental factors such as hypertension, smoking increase the likelihood of IA formation and rupture, these are of limited clinical value in identifying at-risk individuals. And identification of genetic risk factors therefore play an important role in developing diagnostic and therapeutic tools. Well, a genetic risk for IA has been speculated as early as 1960s with a report of 4 families having at least 2 members with cerebral aneurysms. And like any other common trait, most cases of IA do not follow a clearly defined Mendelian inheritance pattern, which is indicating that IA is a complex trait. And recent advances in genomics and genomic technology started to reveal the risk regions and causative gene mutations. But understanding the role of these genes, regions, and implicated cellular signaling mechanisms and how they regulate cerebrovascular integrity needs further functional studies. In this presentation, we would like to provide brief information about the systemic heritable disorders that are associated with intracranial aneurysm formation and rupture, since the clinical management of these patients can be quite challenging. And we know that the prevalence of IA is increased in several heritable disorders, and these are mainly connective tissue disorders. And all these disorders can also cause other types of arterial pathologies, including intracranial dolichoectasias, thoracic aortic aneurysms, or other types of arterial dissections. And therefore, it is important to recognize their clinical presentation to ensure early screening, diagnosis, and treatment. One of the most common heritable disorders associated with IA is, as well known, autosomal polycystic kidney disease, which is a leading cause of heritable kidney failure and hypertension, which is manifested by cysts in the kidney, liver, pancreas. And the prevalence of autosomal polycystic kidney disease in the United States is estimated to be between 1 in 400 to 1,000. And it's caused by the heterozygous mutations in 2 transmembrane proteins, PKD1 and PKD2, which are responsible of approximately 85% and 15% of the disease, respectively. And IA is frequently observed with autosomal polycystic kidney disease with a prevalence between approximately 12% to 22-23%. And in addition to intracranial aneurysms, also several neurosurgical pathologies are associated with autosomal polycystic kidney disease, including arachnoid cysts, chronic subdural hemorrhages, and intracranial dolichoectasia. And given the strong association, radiological screening for IA should definitely be considered in patients with autosomal dominant polycystic kidney disease, particularly those with advanced age and family history. Another group of disorders associated with IA are called brain small vessel diseases associated with type 4 collagenopathies. And these disorders are caused by heterozygous mutations in COL4A1 and COL4A2, which share the same promoter. And COL4A1 and COL4A2 display strong expression in the basement membrane of arteries, but also in the basement membrane of other organs in the body, which explains the systemic and overlapping manifestations of these diseases, including white matter abnormalities, retinal artery tortuosity, intracranial aneurysms, polycystic kidney, and microscopic hematuria, as well as muscle cramps. So the frequency of intracerebral hemorrhage is as high as 11.5%, and unruptured IA is observed as 5.2% in these diseases. And the IA frequency seems to be more than the general population, and it's important to note that there's a high ratio of pediatric patients in this cohort. Although they're named as small vessel disease, interestingly, the aneurysms in these diseases are almost always located in the carotid siphon. Another rare but multisystemic disorder with a prevalence of 1 in 70,000 newborns is Alagille syndrome, and it's caused by mutation in JAK1 and Notch2 genes, which are involved in Notch signaling pathway. Although it's rare, it still warrants the attention of neurosurgeons, since the 25% of the deaths are due to intracranial hemorrhage in this disease. And also approximately 14% of patients develop intracranial hemorrhage. And the prevalence of arterial and venous abnormalities in the brain is estimated 47%, while approximately 1/3 of them are intracranial aneurysms. And interestingly, the majority of IAs identified in Alagille syndrome are found in posterior circulation, and they become symptomatic until late adolescence. Loeys-Dietz syndrome is a systemic connective tissue disorder that shares many overlapping features with Marfan syndrome. And we see heterozygous damaging mutations in TGF-beta receptor 1 to SMAD2, SMAD3, TGF-beta 2 and 3 in Loeys-Dietz syndrome. And we observe a paradoxical dysregulation of TGF-beta signaling in arterial tissues in this disease. And clinically, cardiovascular manifestations, especially thoracic aortic aneurysms, are the most prominent feature. And unfortunately, median survival is 37 years of age, with thoracic aortic aneurysms being the leading cause of death. And vascular manifestations are severe and can actually be observed as early as 6 months of age. And the studies in the literature show that both clip ligation as well as endovascular treatment were successfully performed in patients with ALDS, ranging in age from 11 to 46 years. And however, it is important to note that that these patients may experience increased vasoreactivity and might require additional treatment. Another well-known systemic irritable disorder associated with IA is Ehlers-Danlos syndrome. And Ehlers-Danlos syndrome is a relatively common heritable connective tissue disease with an estimated frequency of 1 in 5,000 individuals worldwide. So 2017 classification recognized 19 genes and 13 clinical subtypes. So we should consider the vascular pathologies in all subtypes, but especially the type 4 Ehlers-Danlos syndrome, which is also called vascular subtype. It is caused by heterozygous COL3A1 mutations and has the highest mortality and morbidity rates due to arterial rupture and hemorrhage at young age. So aortic aneurysms, dissections, ruptures are the leading vascular complications and cause of death in 6 to 8% of cases. And cervical and intracranial vascular events have been reported in as high as 17% of patients. But successful coiling of intracranial aneurysms and carotid cavernous fistula are reported. I would like to talk also about the approaches to the problem of identification of genetic risk factors in IA pathogenesis. And similar to most common disorders like hypertension, diabetes, or neuropsychiatric disorders, obesity, also intracranial aneurysms are considered as common complex disorders. That means that both rare and common variants are implicated in pathogenesis. And one general model for explaining the genetic susceptibility to common complex disorders is the common disease, common variant hypothesis. And in this model, aggregation of large number of common variants, which contributing a small risk by themselves, are responsible of the genetic predisposition. And this hypothesis forms the basis of genome-wide association studies. And over the past 10 years, we and other groups using GWAS provided important insight in IA pathogenesis by identifying several regions associated with IA risk. However, GWAS are not designed to detect rare variants with large effects in sporadic cases as well as familial cases. So therefore, a complementary model, a complementary approach is required to identify the remaining risk, the remaining genetic risk in IA pathogenesis. And this complementary model is common disease rare variant hypothesis. And the susceptibility loci identified in GWAS typically have low odds ratios compared to rare variants, such as like the mean odds ratio for common variants is 1.36 versus 3.74 for rare variants. And recent advances in genomic technologies, including whole exome sequencing, has transformed the field of human genetics. And also, we started to see the result of this application in intracranial aneurysm genetics as well. And using this technology, a few studies demonstrated association of the rare variants in genes, including RNF213, which is also associated with moyamoya disease, THSD1, and angiopoietin-like 6. In this presentation, I try to summarize systemic inherited disorders that are associated with IA and briefly talked about the genomic approaches that are being used to elucidate the underlying molecular biology of intracranial aneurysms. Recent advances in perioperative management of intracranial aneurysms and subarachnoid hemorrhage decreased the mortality and morbidity rates. However, it is important to remember that still as high as 1/4 of patients with subarachnoid hemorrhage die before even reaching hospitals. This emphasizes the importance of developing better diagnostic tools to identify the patients at risk. Therefore, elucidating the genes and the cellular pathways involved in IA pathogenesis and understanding how the dysregulation of these pathways translate into structural changes in cerebrovascular network as well as changes in hemodynamics is not only of intellectual curiosity, but also of clinical importance. Understanding the impact of these genes and pathways on regulation of cerebrovascular hemodynamics, mechanosensation, smooth muscle cell and endothelial cell phenotypic transition will help us develop novel diagnostic and therapeutic tools in the near future.